Hello. This is a summary of the short-term scientific mission supported by Actinpak. The objective of this mission was the development of novel active edible packaging with antimicrobial random peptides as food preservative. This project was born by the collaboration of two institutions. The University of Modena and Reggio Emilia in Italy, expert in edible packaging and food microbiology, and the Hebrew University of Jerusalem in Israel, expert in the synthesis of antimicrobial random peptides. The outlines of these studies were: 1. The synthesis of 20 amino acids random polymers. The amino acids used were phenylalanine, leucine and lysine for one polymer and lysine and tryptophan for the other one. 2. Find and create an optimal formulation of edible food packaging with antimicrobial random peptides included. 3. Test in vitro the antimicrobial power of this active edible packaging. 4. Use this formulation to create an antimicrobial coating on minced kosher meat. The method used to synthesize the random peptides is the one developed by the Nobel Prize Robert Bruce Merrifield. This is a solid phase synthesis, an accurate and elegant way to synthesize peptides. This seeks permit to synthesis of natural peptides which are difficult to express in bacteria. The incorporation of unnatural amino acids. And the synthesis of D proteins, which consist of D amino acids. The general principle of solid phase synthesis is the repetition of this cycle. Deprotectio. Wash. Coupling. Wash. The free N-terminal amine of a solid phase attached peptide is coupled to a single N-protected amino acid unit. This unit is then deprotected, revealing a new N-terminal amine to which a further amino acid may be attached. The superiority of this technique partially lies in the ability to perform wash cycles after each reaction, removing excess reagent with all of the growing peptide of interest remaining covalently attached to the insoluble resin.
After the synthesis of WK and FLK peptides, these compounds were analyzed with a MALDI-TOF instrument to check the mass. The range of the mass are correct for both peptides. The higher number of peaks in FLK chart was due to the composition of this one. FLK had 3 amino acids, so the possible combination during a random synthesis were more.
The film-forming solution was created adding pectin into a solvent. The tested solvents were distilled water added with different concentrations of antimicrobial random peptides and glycerol as plasticizer. Pectin was added while stirring and heating. Then the solution were poured on a 30 mm Petri dish, and calcium solution was sprayed over the gel to reticolize it. The drying process was carried out at room temperature for 24 hours inside a microbiological sterile hood. The dried films were separated from their support and utilized as discs for Kerber diffusion test. The film obtained look like a normal plastic film, stretchable and transparent. It is totally biodegradable and edible. The antimicrobial random peptides as antimicrobial make the active part of the packaging in synergy with the polymer. It is possible to observe how the antimicrobial random peptides included inside edible pectin film can inhibit by contact the growth of E. coli and B. subtilis. In some case where the antimicrobial random peptides concentration was higher, we obtained an inhibition zone by diffusion as well. It is possible to say that the new formulation of active edible packaging showed a great antimicrobial activity. The minced kosher meat was a selection of beef sirloin steak. The treated minced meat was divided in meatballs of 10 grams and coated with the film-forming solution. The meat was stored inside the fridge at 4 degrees Celsius. Each day of analysis, 3 meatballs for each kind of thesis are analyzed. The counting of mesophilic aerobic bacteria was determined respecting the Microbiological Protocol, ISO 4833. After 24h of incubation at 30 degrees Celsius, the bacterial colonies were counted to determine the CFU per gram of mesophilic aerobic bacteria. It is possible to observe 1 log reduction in the mesophilic aerobic growth in all the sample treated with coating FLK and WK random peptids. The quantity of antimicrobial random peptids used in these studies was very low, 5 mg per ml of film-forming solution of pectin. To conclude, it is possible to say that these novel active edible films can reduce the spoiling bacterial population on kosher meat, increasing the shelf life and the food safety. Only one kind of edible polymer was used, so in the next future it will be interesting to see the behavior of antimicrobial random peptids inside other kinds of food packaging. On the other hand, it will be interesting to study the mesophilic aerobic bacteria trend using a higher concentration of antimicrobial random peptids inside the edible film and test it on other food matrices as well. Thank you for your attention and thanks to Actinpak for his support in these short scientific mission.